¿Le avisas?
¿Quieres que le diga? Sí. Alex, you can share the link, the Zoom link, if you want the people
Thanks.
that you wanted to listen to the talk right now. You can share the Zoom link, but they're only asking us to enter in silence and probably the video out.
I've seen one already joined, so I know she's there, Aline. So okay, good. So you just let me know when I start, and then so I talk for 30 seconds and then translate.
Oh, don't worry. First, I'm going to introduce you first in Spanish, and then I'll let you know. Okay.
Okay, perfect.
Buenas tardes, tengan todos ustedes. Es un gusto para mí poder estar con ustedes el día de hoy en la última conferencia magistral. Tenemos hoy un invitado especial, es el doctor Alexander Tubbs. Él va a dar la presentación en inglés, tenemos la traducción y las preguntas, por favor, en cuanto tengan las preguntas las pueden poner en español o en inglés como ustedes quieran y yo se las voy a pasar al doctor. Entonces, para comenzar con ahora sí que vamos a comenzar a terminar con la última presentación. Es un gusto contar con la participación del doctor Alexander Tubbs en este Foro Internacional de Investigación y Posgrado. Él es biólogo egresado de la Universidad de Marburgo en Alemania, en donde también hizo su doctorado en colaboración con el Instituto Rowe de Nutrición y Salud en Reino Unido. Actualmente es profesor asociado en el Departamento de Fisiología en la Escuela de Ciencias Biomédicas en la Universidad de Otago. Es autor de múltiples publicaciones, ha recibido diversas distinciones a lo largo de su carrera. La reseña completa la pueden ver en el perfil del foro. Su interés principal en los últimos años es la regulación neuroendocrina del metabolismo. En particular, quieren entender cómo interactúan las señales de adiposidad, leptina, insulina en el hipotálamo para poder mantener la homeostasis energética, que es básicamente el metabolismo de glucosa, insulina, etc. Y su objetivo es identificar mecanismos que sean novedosos y al final tener productos naturales. Van a ver que tiene varias patentes y también productos farmacológicos para el control neuroendocrino del metabolismo de la glucosa. Entonces, el día de hoy nos va a presentar el papel de la inflamación hipotalámica en la enfermedad metabólica. Entonces vamos a comenzar con la presentación. Alex, please, welcome.
Buenos días, gracias por la invitación a esta reunión. Thank you very much for the invitation to give this talk, especially to Andrea, who asked me to talk in Mexico. I wish I could have come in person and not via Zoom. Would have been much nicer, and I love to hear the language. So unfortunately, I don't speak it, so I need to speak in English or in German, but probably you wouldn't understand German either. So we need to stick to English. And thank you very much. And I would like to start my presentation now. So I will talk about the role of hypothalamic inflammation in metabolic disease. And this is just the highest mountain of New Zealand in the background, outside of the Mount Cook area. So all of you will know that if we want to control our body weight, it's a tight balance of the nutrients that we take in on the one hand, so our energy intake, and then the energy expenditure on the other side. I will come back to that at the end of my talk because it might be not that simple as we think this could be. So I will talk about body weight regulation and what we can do, hopefully, potentially, to control it. So we have a huge problem in the world, another pandemic compared to the COVID pandemic, and this is the obesity pandemic. There are 1.6 billion people overweight and 400 million people obese, and this leads to several chronic diseases like type 2 diabetes, hypertension, or cancer, to name a few. And it costs a lot of money. I don't know the numbers for Mexico, unfortunately, but in Australia, for example, our neighboring country here, it's 21 billion per year, or in the US, it's 147 billion. So it can lead to bankruptcy policy of the health organizations and the health systems in the countries. So there have been never more desperate attempts to find anti-obesity treatments. So I would then usually ask you to, I don't know if I can ask you, but it's hard by Zoom, your chance of getting to lose body weight over 5 years is quite difficult because a study came out in 2015, it's quite a while ago now, where they followed hundreds of thousands of people over 5 years. So if you go on a diet, so you go to your doctor and your doctor will tell you you should eat less and do more exercise. So the chance of this to succeed, to lose body weight, is 1 in 210 for men and 1 in 124 for women, so way below 1%. So you could argue then, is our money well invested into the health system if this is our obesity treatment? It's one question I could ask. So it is way more complicated than we think. And even in the most controlled environment, which would be the biggest loser environment, which is a TV show that some of you might have seen, because this is the best way you can do it. You have the best doctors in the world on the TV, and they will follow you, and the whole world watches you how you lose weight. So you will really adhere to the paradigm that you're told to do. And when you look at all the different seasons of The Biggest Loser, actually none of them kept their body weight reduced. Only one person actually. Yes, so one person maintained a lower body weight, whereas 13 of the 14 participants regained their body weight, and 6 are now heavier than they were before. Most of them had a reduced metabolism. So dieting works for the short term, but not for the long term, as you can see, because all of them lost weight in the short term when they were on TV, but after that, then it didn't work anymore. So it's way more complex than we think. To control our body weight is not a choice that we can just say we eat less and we do more exercise, because our brain controls this. And we cannot override the brain's decision in a way. So one important, or probably the most well-studied hormone that's one of my main focus, is the hormone leptin, which is produced by adipose tissue in our body. And then it enters the brain through the bloodstream, and it binds to the hypothalamus, which is the area in the brain that controls body weight and energy metabolism. What it does there, it reduces food intake, it increases energy expenditure, and it also regulates glucose levels in our blood. So the brain controls glucose levels in our body. And so you think, okay, we could treat obesity by just giving leptin, but it is not that simple, because when we get obese, we have more adipose tissue and we produce more leptin, and then you would think we would eat less. But unfortunately, it's not like this, because the brain cannot read the signal anymore. It becomes resistant to leptin, and then the effect of leptin disappears. And about 95% of humans are leptin resistant, and that's why we can't treat obesity that simple, because the resistance in the brain tells us we need to eat more. So leptin does not function anymore. Our homeostat that controls our body weight became dysfunctional. So we are looking into inflammation as the main culprit or reason for this leptin resistance and obesity. What happens is that saturated fats, for example, from excessive burger intake or too much lard, a very bad diet, they can also act in the brain, and I will talk about this in my talk, and they might desensitize the effect of leptin. So leptin acts in the hypothalamus, and it binds to receptors that you find in neurons of the hypothalamus. And there's another hormone called insulin, which all of you will know, hopefully, and that controls glucose levels, but it also controls body weight in the brain. Both of these hormones interact in the brain, and what happens when the hypothalamus becomes inflamed, that is our hypothesis, that then these hormones cannot interact anymore, and this inflammation leads then to diabetes, because the function of insulin is gone and the function of leptin is gone, and we cannot control glucose levels anymore. And obesity occurs, obviously, because leptin controls body weight and glucose levels. Well, how does it then work? So when we ingest these saturated fatty acids, so the main driver of this inflammation are long-chain saturated fatty acids. One of them is called palmitate. So if we eat too many burgers, what happens? Well, these fatty acids actually enter the bloodstream because we digest them, and then they can go to the brain and bind to receptors that are expressed on neurons. And one of those receptors is called Toll-like 4 receptor, which is the main pro-inflammatory receptor on all cells. And these saturated fatty acids can directly bind to these receptors and then induce an intracellular signaling cascade. The binding of these is not 100% confirmed. It's a bit controversial. There are some papers that show that it's not the case, but what is known that they drive this inflammatory cascade in the cell, which involves these mediators. I will talk about this cascade in a moment. Another way is also that we become the leaky gut hypothesis. So these certain bacteria can enter the bloodstream, and the bacteria have surface polysaccharides that can then induce this chain. So either the fatty acids directly or a change in our bacteria in our gut can drive the inflammation in the brain. And we have then two inflammation cells in the brain, types of cells. One is called microglia. These are the macrophages of the brain. And then another one, it's called astrocytes. Sorry, it's always moving here. So these are the drivers of inflammation in the brain. So we can induce microglia, the macrophages, or we induce astrocytes. And they then feed back to the neuron, and then we have an inflamed state in the brain. So how does it work in detail? So I mentioned already that saturated fats can bind to the Toll-like 4 receptor and then induce this intracellular cascade. This involves two molecules. One is called IKK beta and the other one is called NF-kappa B. NF-kappa B, which you can see here, can bind to the nucleus, and then this induces the inflammation. Certain cytokines, pro-inflammatory mediators in the body, are released by cells and they cause this inflammation. So what you could do, because this little molecule IKB alpha that resides in here prevents NF-kappa B from going to the nucleus. So usually when the saturated fatty acids bind, this is degraded IKB alpha and NF-kappa B can go to the nucleus. So what we did, we thought we can overexpress IKB alpha genetically, produce more of these molecules, and then NF-kappa B cannot go to the nucleus. So it resides outside of the nucleus and you don't get inflammation. So that's how we manipulate inflammation. So usually when it's activated, it induces cytokines and leads to diabetes, for example. So we did this. We wanted to know if is really hypothalamic inflammation the driver for obesity and diabetes. So we blocked hypothalamic inflammation with this technology, and Jonas Benzler, a PhD student in my lab in Germany at the time, designed a viral construct. So what he did, we used mice, and he overexpressed this little triangle that I showed you, this little molecule in the brain of mice. And you can see here, we injected this into the hypothalamus. The hypothalamus would be here, so this is the coronal section of a mouse brain. And here you see these dark dots. These are the overexpressed molecules that we can detect, so it worked. And we brought these molecules into neurons under a neuronal promoter in a way, and overexpressed IKB alpha in neurons. So we blocked inflammation in the brain. So what happens to mice when you block inflammation in the brain? So this is what you see here. So we feed mice different diets. So you feed mice a normal diet or a high-fat diet that is enriched in these saturated fatty acids. And then when you overexpress IKB alpha, so you block hypothalamic inflammation, you see that the body weight increase is lower. So when you put them on a high-fat diet, you see that they gain weight, as you can see here. And when you have them on a normal, after some time here, you see they increase weight. But when you overexpress IKB alpha, this increase is blunted. When we look at fat mass, it has a dramatic reduction in fat mass, but the lean mass, the muscle mass, has not changed. So this is really important to know. So you can reduce your fat mass, but not your lean mass by blocking hypothalamic inflammation. We don't want to lose muscle, obviously. So what happens to glucose levels in the body? So we looked at fasting blood glucose, and about 100 milligrams per deciliter is the decision where you turn into the diabetic range. So the mice in the control condition were diabetic because to feed them a high-fat diet, they gain weight and they become diabetic or glucose intolerant. And by blocking hypothalamic inflammation, you improve this. And this is a glucose tolerance test. So we inject glucose into the body. Glucose then leads to an uptake of insulin. Glucose is taken up into the cells, and insulin is produced so that glucose is taken up into the cells. And this is over time. So you inject it, and then glucose levels drop because insulin is produced. And you see that when you overexpress IKB alpha, so you block inflammation in the brain, you see an improvement in glucose tolerance. And this is insulin tolerance. So we look at the response to insulin, and that was also improved over time. So to wrap up, hypothalamic inflammation, when we inhibit it, it improves glucose and insulin tolerance, and it reduces body adiposity. So it protects from those metabolic abnormalities. So then we thought, can we induce hypothalamic inflammation with certain dietary components? And it seems to be a bit of a jump, but we thought, okay, when we reduce hypothalamic inflammation, we can treat obesity, but do we have things in our diet that can cause hypothalamic inflammation? And New Zealand uses a lot of gluten. So when you go to the supermarket, you can buy gluten as a product, which I haven't seen before. So you can just buy gluten and add that to everything you cook because it makes it more fluffy. And there's a high prevalence of celiac disease here and gluten intolerance. And it's very controversial. Gluten is a very hot topic because there are a lot of gluten-free products that many people may not need to eat. And it's very hard to study in humans because people have some opinion about gluten, and you will know if you have a gluten-free diet or not. And there's the so-called nocebo effect. So people will have an adverse reaction to eat gluten because they anticipate a bad outcome. So mice would not have this because they don't know what gluten is, I hope, and that's why we studied gluten in mice. The effects of gluten, and we wanted to see, does that cause hypothalamic inflammation? So we designed a diet where we added gluten to this diet, and we added 4.5% wheat gluten, and the mice got 4 diets. So a normal diet, normal fat content, which is 10% fat, or 60% fat, which we call a high-fat diet. And then we added to those 2 diets, we had no gluten in these 2 diets, and then we added 4.5% wheat gluten to the other 2 diets, as you can see here. The main factor of fat is derived from lard, which is, again, polyunsaturated fatty acids, saturated fatty acids. So what we see here when we look at body weight and we add gluten to the diet, first of all, when you feed them a high-fat diet, this is the control condition without gluten, and you have the high-fat diet mice, you see a dramatic increase in body weight. So the high-fat diet makes them obese, dramatically obese, like it would happen in humans if you would eat 60% of fat. But when you add 4.5% of gluten to this diet, in mice on a normal diet, you don't see any effect on body weight. But in mice that were already fed a high-fat diet, you see a further increase in body weight. It's a mildly obesogenic effect, so it's not a massive increase, but there's a slight increase in body weight that was significant. And when we look and analyze this effect from the beginning to the end of the study, that's the body weight gain over the whole study, and you see that there is a significant increase in body weight gained when you add gluten to a high-fat diet. Also, when we look at the caloric intake, you see that the calories are increased. They eat more on the high-fat diet rather than on the low-fat diet. And obviously, when you feed mice a high-fat diet, overall, they eat more as well, obviously. So this was interesting. And then we had a look at the different fat depots, because we know now that we had a slight obesogenic effect. So mice gained weight with gluten on the high-fat diet, and we wanted to know where is that body weight gain. So when you dissect mice, you have different fat depots, and we found that in the visceral fat, like in the perirenal, inguinal, and retroperitoneal white adipose tissue, we found an increase by gluten from high-fat diet without gluten and high-fat diet with gluten. So in these mice that were supplemented with gluten when they were fed a high-fat diet, we saw an increase in adiposity. Well, how does it look when we look at inflammation? So we measured CRP, C-reactive protein, which is a general marker of inflammation. So when you go to a doctor with any disease, they will most likely measure CRP. It's a marker for infection. It's a marker of chronic disease. If you have arthritis or something, CRP levels might go up. And what we found, which was quite interesting, so when we add gluten to mice on a low-fat diet, what you can see here, we see quite a significant induction of CRP levels in the blood of those mice, but not in the mice on the high-fat diet that gained the body weight. So it was a bit surprising, but we found in one condition, we found an effect of gluten. So now I would like to come back to the brain. So how does the brain of these mice look like? And I mentioned that we measure hypothalamic inflammation, and I mentioned to you that we have two different cell types. One are the astrocytes, and one are the microglia, the immune cells of the brain. So do we see a difference in those immune cells? And that's what we did. So we do immunochemistry. So we cut the brain into sections, and then we stain for certain proteins. And we look at proteins that are markers for astrocytes and markers for microglia. And one of these proteins is called GFAP, which is a marker for activated astrocytes. So when you find more of those cells, a higher number, then you have more inflammation, because these drive inflammation in the brain. And this is the area that controls body weight. It's called the arcuate nucleus of the hypothalamus. It's a sub-area in the hypothalamus. And then we count the number of those cells. And this is shown in the bar graph on the right. So you see mice on normal diet with and without gluten, so without gluten, with gluten, and then mice on a high-fat diet without gluten and with gluten. First of all, what we expected to see was that the high-fat diet induces the number of astrocytes, which we have seen before, because we know that the high-fat diet induces hypothalamic inflammation. And this is from here to here, so you see that increase. What was very interesting is that adding gluten to the low-fat diet mice leads to the same increase than feeding them a high-fat diet. And when you add gluten to the high-fat diet mice on top of the high-fat diet, you see a further increase. So gluten induces the number of astrocytes, and it does the same to the number of microglia. So these are the macrophages of the brain. Each of these little dots is a cell that is a macrophage of the brain, let's say. And this is the number counted again. It's less profound, but it's also significant. So we see an increase by gluten in both mice on a low-fat and a high-fat diet. So now I would like to summarize these data on gluten. And we can see that both in mice on a low-fat and a high-fat diet, gluten increases inflammation in the brain. So gluten can be pro-inflammatory, we can conclude, in mice. So we cannot say this in humans yet. We can talk about this later, maybe in the discussion. So we know now that we have pro-inflammatory potential dietary components, which might be gluten, and we know that hypothalamic inflammation drives obesity and diabetes. So can we find anti-inflammatory components in our diet? And can we find out what these components actually do? How do they cause this damage. So the main driver for hypothalamic inflammation are long-chain saturated fatty acids, and the main dietary long-chain saturated fatty acid is called palmitic acid or palmitate. You find this in burgers and in lard. It's the main saturated fatty acid. Then, of course, we have healthy fats, and these are fats you find mainly in fish. And this is, for example, docosahexaenoic acid or DHA, which is an omega-3 fatty acid that is supposed to be healthy. Everybody knows that fish and olive oil should be healthy for us because they contain a lot of DHA, for example, or other unsaturated fatty acids. So what is interesting is when we look at palmitate itself, so this long-chain saturated fatty acids that you find in burgers, that can bind directly to this toll-like receptor and induce this inflammatory cascade that we manipulated and could show that this blocks inflammation if you block this cascade and reduces body weight. So we know that about palmitate. What we also know is that insulin and leptin act in the brain, and they have different pathways. Sorry, I need to go a bit into signaling cascades now. I don't want to talk too much about these molecules. This is just to show you insulin binds to its receptor, which is expressed in neurons in the brain. And then you get an activation of the insulin receptor substrate. It's called PI3 kinase and then AKT. So this is how insulin acts in the brain and controls glucose levels by activating this cascade. Then we have another cascade that involves another pathway called the Wnt pathway. And what is interesting with this pathway is that we could show that this is also involved in body weight regulation, but this is not, I don't talk about this today. This was just shown in other studies. So when you change GSK3 in the brain, when you overexpress it, for example, mice gain weight. But GSK3, which is also activated by insulin because it's downstream of this cascade, is the main driver for neurodegeneration. So GSK3 phosphorylates certain proteins that are the hallmarks of Alzheimer's disease. So GSK3 is the main driver of the accumulation of amyloid plaques in the brain, which is one of the main theories of Alzheimer's disease. And it can also lead to hyperphosphorylated tau and then microtubule breakdown. And this is shown here as well, because you have these microtubules, for example, in the brain that then break down when they're hyperphosphorylated. And the third hypothesis of Alzheimer's disease is inflammation of the brain. So we have a direct effect of inflammation in the brain that then can cause Alzheimer's disease. Alzheimer's disease is termed type 3 diabetes for the reason being that it's actually starting with insulin resistance. So it's a type of central insulin resistance where the brain cannot respond to insulin anymore. And you can treat Alzheimer's patients in early stages by giving insulin. So we wanted to see, does the diet have any effect on any of those markers? And maybe that's how it causes inflammatory effects and then the breakdown of certain cell types that control body weight, for example. So that's what we did. We wanted to know which dietary components play a role in Alzheimer's disease, and maybe by changing neuronal function and structure, they also impact our body weight. And then Arlene came on board, which she was a PhD student. Here she flew to New Zealand, and then COVID arrived, and then she left New Zealand 2 years ago. So we still have ongoing results to do. So if somebody wants to come to New Zealand and finish this work, you're welcome. So she used cell culture because it's very difficult to give individual fatty acids to an animal. So you cannot just feed palmitate. So we wanted to see what is each of these individual fatty acids, what are they doing? And it's much easier in cell culture. So we took red pups and we took neurons of these red pups where Alzheimer's starts, for example, in the cortex and in the hippocampus, and then we dissociated them and we treated them with these fatty acids to see what happens to them. And we did different measurements. And this is what you get. So these are neurons in the brain. They look beautiful when you check them under the microscope. And these are healthy neurons on this panel here, we use different markers to stain them, and this stains mainly the axons. So neuron has the cell body, and then the processes, we call them axons and dendrites, as you can see here, the dendrites and the axons. How via these they communicate with each other. When you treat them with palmitate, they look not so nice anymore, so they look very different. So that was interesting, and this looks very similar to this breakdown of the cytoskeleton that you see with this hyperphosphorylation of tau, or these neurofibrillary tangles, how it's called. So this is one of the hallmarks of Alzheimer's disease that we see when we treat them with palmitate. What happens if you give them fish oil or DHA, so the good omega-3 fatty acid? It had no effect. So they look very healthy. And then we did an experiment where we treated them with palmitate and DHA, and we see that DHA, omega-3 fatty acid, prevents the effect of palmitate. So here you could say, okay, it's just toxic. Palmitate is toxic to the neurons and they die. But interestingly, if you treat them with DHA on top of it, this toxicity is gone and they look healthy. And this is just the quantification. So we measure the cell body size as a marker, and here you see palmitate increases the cell body size, DHA has no effect, and it prevents the effect of palmitate. So what causes these morphological changes then? So this is Arlene here, she asked this question, and we had a look at the cytoskeleton. And when we look at axons, in the axon, you find microtubules that maintain these axons, and that's how neurons can transport substances through their cytoskeleton. When you look at the microtubules by measuring beta-tubulin by immunocytochemistry, you see they look very different again, and DHA prevents this effect. What happens to microtubules? They can destabilize by these tau proteins that dissociate. That's a hallmark of Alzheimer's disease. Or they can even hyperstabilize when they're hyperphosphorylated, for example, which is another hallmark of Alzheimer's disease. So they are just not looking very healthy. And this is exactly what happens when we treat them with palmitate. So it leads to breakdown of microtubules. And this was measured by, we just looked at the axon thickness, the process thickness over the length of the axon, and you see that palmitate leads to a much thicker axon, which is an indication that the microtubules are not healthy anymore. And DHA can prevent this over the whole length of the axon. So it seems to be beneficial to take DHA on top of palmitate. The primary process per neuron and the branching events did not change. So just the thickness of the axons. So then we looked at the axon transport. So substances are transported through the axon, and we wanted to see if that's impaired by palmitate. And what would happen then is that, so the question is, are synapses affected by cytoskeleton alterations? Because at the end of the axon, you find the synapse. That's how neurons communicate with each other. So we wanted to see, are these synapses affected maybe? Do we see a different number of synapses? So this is the end terminal, and then substances are transported through the neuron, neurotransmitters, for example, and they are released. And that's how neurons communicate. And if you have less of these connections between neurons, you have less communication in the brain. So to do this, we stained them with the synaptic marker synapsin-1. And all these red dots here are synapses. So these are contact points between each neuron, so they contact each other, and that's how we make our memory by connecting these neurons to each other. And you see that palmitate reduces the number of synapses. DHA had no effect, and again, DHA and palmitate actually works preventative. So here, this is the quantification. The number of synapses was nearly halved by palmitate treatment, DHA had no effect, and when you give DHA plus palmitate, this effect of palmitate was gone. And to conclude the final experiment, we looked at the number of astrocytes again. Remember, these astrocytes were changed by gluten. Gluten induced the number of these immune cells in the brain, and also palmitate does this. So these are astrocytes in the cell culture, and you see that the number of astrocytes induced by palmitate, and they look also brighter, and DHA prevented this effect. They also look less healthy. So to wrap up this part of the talk, so palmitate modulates neuronal morphology. It changes cell body swelling, increases cell body swelling, blabbing, and it reduces synaptic innervation. These morphological changes are caused by the alteration in the microtubules cytoskeleton, as I could show you. The microtubules look different, similar to Alzheimer's disease. And we didn't see any changes in membrane alterations. I didn't show you this part of the talk. So it is more like the microtubule cytoskeleton that's affected. And all these changes can be prevented by giving fish oil or DHA. So this was very interesting. So we wanted to know next, what causes these changes? So is the insulin pathway that I could show you maybe changing these neurons? So what we did, we actually blocked the insulin pathway and we blocked this molecule GSK3 to see if that affects the effect of palmitate on neurons. When we block GSK3, because that is the enzyme that leads to phosphorylation of tau and amyloid plaques, we were surprised that we didn't see any effect. So this was at different time points. So you see palmitate leads to an induction in cell body size, but blocking GSK3 had no effect. And palmitate stood together with GSK3 inhibitor, this was a pharmacological inhibitor, was not different. So there was no effect at these 2 time points. Then another postdoc came on board in Germany who unfortunately passed away 2 months ago at the age of 42. So he did look at phospho-AKT as the marker in this pathway. He measured AKT at the end as a marker of insulin signaling. And he could see that palmitate, this is by Western blot, you see a very rapid induction of palmitate over time. At the beginning, it's much higher, and it induces the insulin pathway. So that was a surprising result we had. So palmitate induces insulin signaling in the brain. So it acts like insulin. So then it was obvious that we need to block this insulin pathway, and that's what we did next. So we used very specific inhibitors for this insulin pathway. Insulin pathway inhibitors, they were designed in Auckland by a collaborator, and these are they're just different compounds, they all do the same thing. And what you see here, you give palmitate, it induces, again, cell body swelling. When you block the insulin pathway, here together palmitate with these blockers, you block the effect of palmitate. So it looks like palmitate induces these morphological changes by overactivating the insulin pathway. And preventing those changes, we need to target the insulin pathway and reduce insulin signaling in the brain rather than overactivate insulin signaling. So it's all about homeostasis. You don't want too much insulin function in the brain. Okay, so now I would like to show you the last experiments we did, and these were very New Zealand specific. So this was cell culture that I showed you, and we wanted to see what happens if we can treat animals with palmitate, so a lard-enriched diet, but also with a diet that has DHA. So can we give them a specific DHA, omega-3 fatty acid enriched diet? And the reason for this was that actually some papers came out that showed that the diet composition might play a role in body weight regulation. And I come back to this at the end of my talk. So what we did, we fed mice for 1 week and a longer duration for 1 year as well. We fed them a low-fat diet and a high-fat diet. And this high-fat diet I showed you already has 60% of calories mainly from lard, so saturated fatty acids. Our omega-3 fatty acid enriched diet contained a New Zealand product that you can only find in New Zealand, and this is the green lip mussel. It is one of the most unique products in the world in terms of its omega-3 fatty acid composition. It has very high levels of DHA, but it has also very rare omega-3 fatty acids that could potentially have very beneficial effects. We don't know exactly what these fatty acids are doing, but we designed a diet that contains parts of the high-fat diet by lard. So we have three groups of mice. We have low-fat diet, high-fat diet. All this gray stuff is lard. So more than 50% of the diet contains lard. And then they still have a lot of lard here, but a large chunk of the high-fat diet is actually oil. So the fat composition of the green lip mussels. So calorically, these diets are identical. So both of them have 60% fat, but different fats, obviously. Then we fed these for 1 year or 1 week, where different experiments. We fed these mice with these green lip mussels, and we had different concentrations. So this was the highest concentration. We call it the M4 concentration, where we have like all of this is green lip mussel, and then lower concentrations. And then we had fish oil as well. We wanted to compare it. Is it better than fish oil? Because fish oil everybody knows, and this was a New Zealand fish as well, a hoki fish. Many of these fish capsules are made from those as well. Again, we had a high-fat diet, all of these high-fat diet had 60% fat, but the composition was different, and there were different levels of green lip mussel or different levels of fish oil. So what happens to body weight? So this is the low-fat diet condition, and then we give them the different concentrations. So we give them high-fat diet and then different concentrations of muscle oil. And you see that the highest concentration did not lead to any body weight gain. So the high-fat diet leads to body weight gain, whereas when you give them a high concentration of green lip mussel, it does not lead to body weight gain. Fish oil does. Even the highest level of fish oil did lead to body weight gain, as you can see here. So it's still different to there. So this was not different significantly. And that's what you see here. So this is the body weight change from the beginning of the experiment to the end of the experiment. And you see all of them gained weight, except this one, which was the one that had the highest green lip mussel concentration. So well, maybe the mice don't like the diet, they don't eat it, and that's why they lose weight. It's quite simple. So you just don't like something and you don't eat it. It's not that simple. They love the muscle oil diet. So they eat exactly the same, like the mice that are fed lard or the mice that are fed fish oil. There's no caloric difference. So they eat the same amount of food, but they lose weight. And then we had a look what happens when you take mice and make them fat, and then you give that. Does it help to lose them weight? So we fed them for 1 year on a high-fat diet, so they got really, really fat. And then we gave it for some time, and you see that they lose body weight. So you give them this M4 diet, the most concentrated muscle diet, and they lose their body weight. So this is the high-fat diet body weight gain, and then you turn them back to a normal diet. So they lose body weight on the normal diet, and they do the same when you give them a high-fat diet that contains muscle oil. And this is just the body weight differential again, as you can see here. And so they lose this body weight that was different to the high-fat diet control. And there was no difference, again, in food intake. So you can lose body weight by eating a lot of these mussels, but you don't eat less. So the caloric intake is the same. There was no change in energy expenditure. And when we looked at the adiposity, we see, again, we measured the body fat pads, and you see that there were differences in certain fat pads. So total fat mass was partially reduced. And we found partial reduction in certain fat depots in the mouse's body. So to come back to this diagram. So it's not that simple. It's not a simple equation of what you eat on the one hand and what you burn on the other hand, not in terms of energy. So you can have the same energy intake, 60% of fat from lard or 60% of fat from green muscle oil and lard. So you also have bad lard still in the diet, but you have a massive difference on your body weight, at least the mice do. So it depends on the quality of carbohydrates, protein, and fat potentially. So the quality of fat rather than the quantity plays an important role. So I can't tell you anything about the morphology of these brains of these mice because we need to analyze them and I need more students to do this. So if somebody wants to come to New Zealand, please do so. And I would like to wrap up now. So dietary gluten induces hypothalamic inflammation in mice. I could show you this. Palmitate causes neurological changes in neurons that are similar to Alzheimer pathology. So when we looked at these brains, they looked very different. These neurons, we had these changes that look like these amyloid plaques and the neurofibrillary tangles. All of these changes could have been prevented by DHA in mice or in these neurons. If you give DHA on top of it, palmitate had no effect. In a green muscle enriched high-fat diet, protected from obesity in mice. So we can potentially conclude, eat less gluten and more green muscle for metabolic and neuronal health if we can translate this to humans. What I want to say about the gluten experiment as well is we didn't give them anti-inflammatories, for example. If you eat more gluten, it could lead to hypothalamic inflammation, but if you have a healthy diet otherwise that has a lot of anti-inflammatories, polyphenols from plants or green muscles, for example, then the effect of gluten might not be happening. So we need to test this in mice, for example. That would be one of these next experiments. And at the end, I would like to thank you for your attention. Gracias. And I would like to thank my team, all the people that I acknowledged already, and the rest of my team, and Aline Löfhelm as well, who left. And yeah, thank you very much.
Thank you, Alex. It was a very, very interesting talk. I have several questions, but I'm going to first let some other people participate. We have a question, and it says, could you comment on the consumption of fatty fish DHA content versus its content of methylmercury and other contaminants?
Yes, yeah, good point. So this is analyzed. So we get these from a company here in New Zealand, in the Marlborough Sounds, that nice image with the dolphins, and they analyze the mercury content. It's very low. So I don't know exactly. I have the quantification somewhere, but it's extremely low. They cannot sell it otherwise. So both of that was controlled. So it was not in relevant levels. And cadmium as well, no heavy metals in, yeah, all of the batches are measured.
Okay, great.
Good question, though. Yeah.
We don't have any more questions, but I do have one. We have been studying the role of micronutrients in obesity and micronutrient deficiencies in obesity and inflammation.
Yeah.
We've found that because of the diet, the kind of diet that obese people have, they have micronutrient deficiencies, and some of the micronutrients are very much related with inflammation pathways.
Yes.
And some of them are also very important for brain function.
Yeah.
Such as iron and zinc. And so have you thought of exploring this, or have you even done anything related to micronutrients such as zinc that are also related with insulin and leptin? I mean, they're everywhere.
Yeah, it's a good question. You mean trace elements more, or like zinc?
Yeah,
or...
it could be trace elements or even vitamins. Some of the vitamins that are vitamin C or vitamin E that have been associated with leptin, and also they're potent antioxidants. So
yeah, we do a lot of work on polyphenols, for example, antioxidants, and we found different things. So for example, naringenin, which is an antioxidant that you find in grapefruits, was actually not so good. It induced inflammation in hamsters, but then we found others that were highly anti-inflammatory, and butene, for example, was one, and that improved inflammation in the brain and improved glucose tolerance as well. So we work with these polyphenols. I haven't really worked with trace elements, but that would be interesting. And vitamins as such, we haven't given them, but you could give them into the brain. The question is how much of them actually enter the brain as well. That's not so well studied because there's a blood-brain barrier, and some of the vitamins might not enter the blood-brain barrier if they're very big or complex.
Yes, I agree. Maybe trace elements.
Yeah, trace elements would be better. Yeah, they would easily go. Yeah.
And now you finished your talk with saying about it's more about quality than quantity in terms of fat, and I think that's a very, very important message in terms of weight loss, because people are so afraid of fats when they're in a weight loss regimen or diet or anything, that they just avoid eating any fats at all. And we know that that's completely the opposite. And for example, in Mexico, we have avocado that it's very good and it has very, very good fats. So it's also the quality of fats. So my question would be, now that you're exploring this, and a lot of the people here are dietitians and nutritionists that would like to translate your message with the patient, what would that message be?
I must say, we did these experiments in mice, but we can learn a lot from mice because their digestive tract and everything is very, very similar. So my message would be, I mean, what we learned in this muscle experiment was very surprising, because you can eat as much. So they eat a lot of fat and they don't gain weight. So their caloric intake was the same, like in another group that had a lard-enriched high-fat diet, but they didn't gain that weight. So my message is that people should eat fat. And when you look at the body weight regulation effects of fat, because you get a higher satiety signal from some fats in the diet, but then the composition of the fat is important. And the message from the DHA experiments would be that you can also eat a certain amount of lard, and that's also what we see in the muscle experiment. They still had quite a lot of lard in the diet, as long as you have omega-3 fatty acids on top. So I think if you have to a certain level, even an unhealthy diet, but you eat a lot of plant-based omega-3 fatty acids, for example, oleic acid, for example, or you eat a fish-enriched diet, you can compensate for some of these effects. So yeah, my message would be not to be afraid of fats, obviously, because that's not the thing that drives up the body weight. It's these fat-free products, for example, that are sugar-enriched that drive up the body weight, because people think they're healthy if they buy fat-reduced products, but they don't make them as full because of these hormones. They are differently regulated by fat and proteins, for example, than by sugars. The release of gastrointestinal hormones, for example, can be affected by this, and the satiety signal is different. So my message would be to be not afraid of fat. Look at the different levels of fat that are taken in, which quality of fat, green mussels or fish, would be beneficial.
Great, thank you very much. And I don't see any more questions. I'm going to switch to Spanish very quickly, Alex.
Yeah.
Si no hay más comentarios, Andrea, no sé si tengas algún otro comentario.
No, yo no tengo comentarios. Está bien.
Okay. So I'm going to switch back to English. Alex, thank you so much for this incredible talk.
Thank you.
It was great. I would love to talk to you a little bit later about the micronutrient stuff that we've been doing. And also you mentioned microbiota, and that's also something that's so interesting, and we can spend hours talking about it. So thank you again.
Thank you.
So I'm going to switch back to Spanish. A todos los que se quedaron hasta el día de hoy, vamos a cerrar el primer día del foro. Me comentan que lo que seguía son los premios, pero en los premios lo que se va a hacer es que las personas que ganaron los premios se les va a llegar un correo personalmente para avisarles quién es la persona que ganó los regalos. Y invitarlos el próximo viernes 13 de mayo va a haber una plática muy interesante que es la plática magistral del doctor Lafrano sobre biomarcadores de vitamina A para determinar deficiencia y toxicidad. Ese es a las 8 de la mañana. Y tenemos todo el día las ponencias del doctorado en ciencias biológicas, maestría en ciencias biológicas y la maestría en gestión integrada de cuencas. Andrea, no sé si quieras decir algo más antes de cerrar.
Sí, bueno, agradecerles la oportunidad de también invitar a Alexander Tops. Yo creo que puede hacer también una buena línea de investigación si les interesa, y creo que está más que deseoso de colaborar. Me ha dicho de cuándo lo van a invitar a México, entonces sería importante también pensar si pueden hacer algo o trabajar con él, ¿no? Tienen la oportunidad de que a él le gusta trabajar con mucha gente, entonces a lo mejor podrían revisar. Y bueno, thanks Alex for everything. So,
Thanks, Andrea.
okay.
Thank you again, Alex. Y a todos los demás, muchas gracias. Nos vemos el próximo viernes y que pasen buena noche.
Buenas noches.